Hi everyone, this little video is going to discuss the structure of a single nucleotide. So first of all, what is a nucleotide? A nucleotide is a building block for nucleic acid. Just like amino acids are the building blocks for proteins, nucleotides are the building blocks for nucleic acids. And nucleic acids, there's two main types, DNA and RNA. So if you're going to build DNA or if you're going to build RNA, you need a whole bunch of nucleotides that you're going to string together in a long line. So what does a single nucleotide look like? So if we go over here, a single nucleotide is this one molecule that I'm circling. So it looks pretty big and complicated, and it contains three components. Nucleotides have a sugar part, which is this hexagony pentagon thing here, a sugar, a phosphate group, which is what this is, and something we call a base. So each nucleotide has a sugar, a phosphate, and a base, and they're all put together in a very specific way. So what we're going to do is look at this sugar first of all. Now this is a 5-carbon sugar. And it's in a ring, and anywhere where there's a changing direction here, there's a carbon. They're not showing you, but there's a carbon here, and a carbon here, and a carbon here, and a carbon here, and even a carbon here where this makes a little angle. And this green pentagon, which is symbolizing a sugar, can either be this molecule here or this molecule here. And so this sugar could be either a sugar that we call ribose, or it could be a sugar that we call deoxyribose. If you're building a piece of DNA, you have to use nucleotides that have this as their sugar, deoxyribose. And if you're building a piece of RNA, you need to use nucleotides that have this as their sugar. So your body has two different types of nucleotides, those that use ribose in this place and those that use deoxyribose here. And depending on what you're building, whether you're building RNA or DNA, your body will use the correct type of nucleotide. All right, so what we're going to do is go around this sugar one by one of all these different positions on the carbon, all these different carbons in the ring, and learn something about each one. It will help you to recognize and identify what's going on in this molecule. All right, so first of all, when we're numbering these carbons around the sugar, we always look for the oxygen first. So here's the oxygen. The pentagon always has an oxygen in the ring, right here. And then we start going clockwise around. So here's my O, and I'm going to go clockwise. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and then carbon number 5 is the one that's stuck up here. It's hard to know there's even a carbon there, but there's just not shown here on this diagram. If we look at this one, here's the oxygen, and we go clockwise. There's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. All right, so let's go back to this diagram up here. All right, so carbon number 1, the first one around the ring from the oxygen. Carbon number 1 always has the base stuck to it. So when you're building a nucleotide and you've got your sugar, on carbon number 1, you're always going to attach a base. We'll come back to what those bases are in the next video. So carbon number 1 has the base attached. Carbon number 2, which is this one here, this is the carbon that's going to tell you whether you're looking at ribose for the sugar or deoxyribose. So if you look at these two sugars down here, they're absolutely identical except for carbon 2. And if you look at carbon 2, on this molecule of ribose, there's an OH, a hydroxyl group right here. And if you look at deoxyribose, which is this sugar, there's just an H, a single hydrogen. And that's why this is called deoxyribose. It's ribose deoxified, or without an oxygen. Everything else in these two molecules is completely the same. It's just on carbon 2 in ribose, you have an OH. and on carbon 2 in deoxyribose you have just an H. And that's the only way to tell these two apart. So carbon 2 is pretty important. So carbon number 1 has the base. Carbon number 2, that's what you're going to look at to decide whether you're looking at ribose or deoxyribose. Carbon number 3 right here, in all cases, doesn't matter which sugar you've got here, carbon number 3 will always have a hydroxyl group. Look, OH if you're using this sugar, and OH if you're using this sugar. And that OH is super, super important. It's probably the most important part of this whole molecule, which you'll understand why in a little bit further down the line. But carbon 3 always has a hydroxyl group sticking down here. All right. Carbon number 4 is the only one that's boring. I have nothing to say about poor carbon 4. Carbon number 5, that's always the carbon that has the phosphate group attached. So now we know something about each carbon in this ring. We know carbon number 1 is where the base is attached. Carbon number 2 is going to tell us whether we're looking at ribose or deoxyribose. Carbon number 3 is always going to provide us an OH here. Carbon number 4, it's kind of boring. Carbon number 5 is going to have a phosphate group attached. All bases have the same layout, the same design, and with those same important landmarks. And that's the basic structure of a nucleotide.